So today, I would like to share some of our research finding on the multifunctional metal nanotexaphyrin as radiotheranostics. So to begin, I want to talk about theranostics. Theranostic in the simplest term is by combining therapy and diagnostic into one single agent. And recently, targeting has been part of the mix of theranostics because of the selectivity and specificity improvement that you can do for the different theranostic agents. And nonetheless, nanoparticles is a very, very important platform that is used in theranostics. For example, you can have a liposomal platform, a gold nanoparticle platform, or polymer platform, whereby you can load different agents to enable therapy, imaging, or targeting. And that kind of brings us to nanoparticle design. One of the most common way and most popular way in terms of making a multifunctional theranostics is through this all-in-one approach. And the all-in-one approach uses the nanoparticles as a nanocarrier in which you can load different drugs or imaging agents into those nanoparticles or label them onto the surface. Although it's really popular, it has some limitations, especially when these synthesis requires multi-steps. And this can induce more complication in the fabrication and the purification process for those nanoparticles. This kind of raised this hurdle for clinical translations and causing issues such as scalability or reproducibility for these all-in-one approach nanoparticles. In the contrast, you have the newer idea in terms of the 1-for-all approach, in which you can use the nanoparticle building block also as the drug, the imaging agent, or the trigger agent. This design is pretty simple, partly because the nanoparticle building blocks can then self-assemble to the desired nanoparticles with different functionalities. But one of the main key with this design is that you have to synthesize the building block that you need for your treatment or imaging. So it comes to what we're really interested in our lab over at UHN. Our lab is in particular interested in a compound known as texaphyrin. Texaphyrin is similar to a porphyrin, but it has a 5-coordinated expanded ring. And this not only allows the texaphyrin to chelate a range of different metals, including the transition metal series, but also larger trivalent ion such as the lanthanide metals. And not surprising at all, texaphyrin has been used for biomedical purposes. And by chelating different metals, for example, gadolinium, you can introduce new functions to these compounds. For example, it has been investigated as radiosensitizers. And similarly, by chelating lutetiums, you can induce photosensitizations into these compounds. So what we're really interested in is whether or not we can use this mix and match approach of different metals in our single texaphyrin phospholipid platform to have different functions, for example, indium-111 for SPEC and lutetium for photodynamic therapy, and then self-assemble them into this all-in-one approach for this nanoparticle then you can modify with different ligands, for example, the prosthetic-specific membrane antigens targeting ligands, so that we can actually have a platform that enables us to do therapy, imaging, and targeting at the same time. So to do that, first, we need to synthesize a texaphyrin-free base phospholipid conjugate. And then we need to chelate this compound with our lutetium using lutetium acetate in a basic condition with mild heating. Upon getting this lutetium texaphyrin lipid, we can then combine that with a mix and match approach to start our formulation development. And by combining it to a 9:1 ratio to our free base texaphyrin alongside with other lipid components such as cholesterol, DSPE, PEG, and a host lipid, we're able to self-assemble a nanoparticle known as nanotexaphyrin. With this nanotexaphyrin here, which is lutetium-free based nanotexaphyrin with the formulation shown on the right, we're able to see some very interesting optical properties. We can see that in both the absorption and the fluorescence spectrum, that there seems to be some quenching that is occurring here. When the nanoparticles are intact, you can see that there is a fluorescent quenching efficiency that is up to 96%. And these nanoparticles can become disrupted in a different environment. And when they do, they can recover that fluorescence as well as other photo function that, that the lutetium texaphyrin lipid exhibit. So the other thing that we looked into is the colloidal characterizations. And we can see that from our DLS data that the nanoparticles are around 110 nanometers in size with a PDI less than 0.2. And this is in agreement with our TM images, as we can see that we have a homogeneous populations of these spherical nanofascicle structure, which resembles to similar morphology as what we previously observed with similar liposomal structures. Using this mix and match approach, the next thing we investigated is the radial labeling using indium-111. With the free-base texaphyrin, we can chelate indium-111 chloride using a proton sponge, which is also a basic oxidative reagent, in ethanol. And we aim to mix that with our co-lutetium and texaphyrin lipid, and then self-assemble it to our desirable indium-111 lutetium nanotexaphyrin. We did that using a platform which is the microfluidic chips. These chips have different channels with herringbone structures that enable rapid mixing when we co-inject our radioactive lipid in ethanol with the aqueous buffer together. And using this platform, we're able to synthesize our nanotexaphyrin with the indium-111 and lutetium. And we're able to see in both ITLC method and radial HPLC that this radial labeling is very efficient with over 90% radial labeled and radiochemical yield. These nanoparticles can also be purified using size exclusion spin columns, and you can remove any excess unchelated indium-111 that way. We also show that after decay, these nanoparticles show very consistent colloidal properties in both the DLS and the TM as we see here. Now having this nanoparticles means that we can start assessing its in vivo ability. So what we did was we intravenously injected these nanoparticles into a subcutaneous KB models. As we can see in those videos that at 30-minute post-injections, we can see the nanoparticle entering the blood pool, and we can see that there is liver and spleen accumulations throughout different time points, which confirms that our nanoparticle probably took this hepatic clearance pathway. And we can also see that over 4, 8, 24, and 48 hours time period, we can see this tumor accumulations at the site, which is pointed with the right arrow here. We further investigate the PK of our nanoparticles using leveraging on this indium-111 radial properties. We found that our nanoparticles have a elimination half-life of 6.62 hours in the healthy biopsy mice model that we use. We can also see from ex vivo gamma counting that the biodistribution and radial depositions of our nanoparticles show that there is tumor accumulation of around 1.97% ID per gram, but with the majority of nanoparticle accumulating in spleen and liver as we expected. And for image contouring, we can see that this data at 48 hours coincide with our imaging contouring data, in which we can see that the nanoparticle retained in the tumor with a peak accumulation at 8 hours. And so the next step for us is to see whether we can introduce this urea derivative to target PSMA. And to do so, we collaborated with the Pompers group in Johns Hopkins Medical School, in which they provided us with this PSMA targeting ligand lipid, in which we formulated our PSMA lutetium nanoparticles, in which we can see that in both the absorption and fluorescence, as well as our data for our colloidal properties, haven't significantly changed based on the introductions of the PSMA lipid. And furthermore, when we looked at the radial labeling, this introductions of the radial labeling method we used previously also did, with the PSMA included in our formulation, also showed over 90% chelation in terms of the radial chemical yield, showing that this introduction of PSMA did not interfere or had causes any issues in the radial labeling process. So with this nanoparticle that now we have the PSMA targeting, we investigated the in vivo targeting aspect in a subcutaneous dual flank model. With the left flank with our PSMA negative tumor, which is the FLU cell line, and the PSMA positive tumor on the right flank, which is the PIP tumors. We're able to find that in both near-infrared imaging and SPECT-CT imaging with our PSMA nanotexaphyrin, that there is a preferential uptake in the PSMA positive tumor over our negative tumor, and that that proves our selective targeting of our nanoparticle with the PSMA, as we can see again in the SPECT-CT images with the right arrow here. And we also perform some studies on the radiopharmaceutical profiles of this PSMA indium lutetium nanotexaphyrin. And we were able to find that the blood clearance, the elimination half-time is a little bit faster than our previous formulations, but overall very similar in terms of the clearance time. But what we can also see with the ex vivo gamma counting for biodistribution is that at 48 hours, we can see that there is a significant improvement in terms of tumor accumulations at our PSMA positive tumor over our negative tumor. And from the image contouring, we harness more information regarding the comparison between two tumors, but also what time point where the tumor accumulation peaked. And we were able to see that at 8 hours in the positive tumors, we were able to get over 4% ID per gram of our nanoparticles based on our image contouring. And with that being the case, we wanted to wrap up the studies with some in vivo PDT experiments. And using the information we obtained from our imaging and gamma counting, we decided that the 8-hour post-injection time point was optimal for us to perform this study. We compared our treatment group, which is the nanoparticle injected intravenously again, and then the tumor subsequently irradiated with laser. And when we compare this group to our free control group, which is nanoparticle only, saline only, and laser only, we're able to see some photodynamic therapy occurring in at the tumor site. 3 days post treatment, we can see some blanching of the area of irradiations, and around 9 days, we can see some scarring that occurs, which will recover after 17 days or so. And when our endpoint of our study at 30 days, we can see that there is significant tumor growth delay against the free control group, which is also reflected in our tumor growth curve, as we can see that there is statistical significance when we're comparing our treatment group against our free control groups. And that also, we don't see any distinctive toxicity from our weight of the mice over 30 days, as well as some histology, which is not shown here, but you can see them if you would like to. So overall, I think we've been able to demonstrate that the metal nanotexaphyrin is a very versatile platform for targeting and multifunction purposes. And by introducing the indium-111 metals into our nanotexaphyrin, we can enable SPECT-CT imaging. And by introducing the co-lutetium-175, we can enable both the near-infrared fluorescent imaging as well as PDT treatment to show tumor growth inhibition. And finally, this nanoparticle is very tunable. So by introducing a small molecular target into our nanoparticle, such as the PSMA target, we're able to see this selective uptick in our PSMA positive tumor as well. So overall, this nanoparticle have shown and demonstrate that it is possible to use this all-in-one approach by tuning different metals to customize a nanoparticle for different biomedical purposes, but also enabling cancer therapy and imaging, and also targeting in this one single platform. So finally, I would like to thank my lab, my supervisor Gang Sang, and everyone who have worked in this project, as well as all the supports from the lab members. And finally, the funding agency that sponsored this research, especially MMIN and Star 21. Thank you very much for listening, and I'll take any questions.
Thank you, Miffy, for the great talk. We do have a question from Urs, actually, a technical question. How does the proton sponge method work to radiolabel the nanotexaphyrins?
So the metal chelation usually needed at basic conditions or oxidative conditions. So in the ethanol case, we use the proton sponge in the kind of same way as we would use triphthalamine to label the metals. So for texaphyrin, before chelation, the texaphyrin is actually not an aromatic ring. And so with oxidation and metal chelations, you can help to get this metal into the the texaphyrin lipid and also aromatize the ring. The exact mechanism is actually unknown in terms of how exactly that happens, but oxidation is an important role and part of that chelation.
Then we have time for one other question. So the nanoparticle showed strong signal enhancement in the liver as compared to the tumor side. Do you have any comment on that?
Yeah, so the nanoparticle is around 100 nanometers or so. And as we know in the nanomedicine field, that these nanoparticles often take the hepatic clearance pathway. So the accumulation in liver and spleen is actually very common. And so despite that we're getting a lot of spleen and liver accumulations, one of the things is the dose that we're using are not very high, first of all that. And secondly, if we're looking at a specific tumor type, for example, we were investigating prostate tumor, we have previously shown that despite you having a strong liver signal, the surrounding tissue signal is actually very low, especially with the targeting ability. So I guess it depends on the application that we were trying to achieve in this case too.
Thank you. Okay, then with this we are perfectly on time.